Hello everyone, let's start Recall Zoology with me. So in the previous video, we have taken the introductory part of the human kidney and the different types of excretory structures present in the kingdom Animalia. Okay, now let's go through the next topic, that is how urine is getting produced and how it is getting concentrated under heading urine formation and the countercurrent mechanism. Okay, so see here, we know like whatever blood is coming for the filtration purpose right from the heart to the kidneys via renal artery, it is oxygenated but it is having urea inside it. So how the filtration occurs and all we will check here. So the urine formation will be taking place in the three different steps: glomerular filtration, reabsorption and the tubular secretion. Okay, so what is glomerular filtration? So whatever blood, just now I told you, the blood which is coming, which is carried via renal artery into the kidney, it is oxygenated and having the urea, and this blood is needed to get filtered. This blood via renal artery comes inside the kidney, and that renal artery will divide into number of arterioles, afferent arterioles. Whatever this afferent arteriole is there, afferent arteriole will be going inside the Bowman's capsule, and that afferent arteriole will be forming the bunch or tuft of the capillary. You could call it as glomerulus. Okay, now here we will check. So how much amount of blood is coming to the kidney per minute and how much amount of blood is getting filtered, and what is that pressure due to which this blood is getting filtered. Okay, so the total amount of blood per minute which is coming to kidney is 1100 to 1200 ml per minute. We know the cardiac output per minute is around 5 liters, means 5000 ml of blood is pumped out via heart every minute. So if you compare that value, whatever this value is there, the amount of blood received by the kidney is 1100 to 1200, in the sense it is 1/5 of the total cardiac output every minute. So whatever 1100 to 1200 ml of blood is coming to the kidney, it is getting filtered due to the pressure in the glomerulus. We could call it as glomerular capillary blood pressure. Glomerular capillary blood pressure. Due to this blood pressure, the blood is getting filtered, and on an average you could say 125 ml of the filtrate is getting produced in the Bowman's capsular space per minute. Okay, so if we calculate in that way, definitely 180 liters of the initial filtrate urine will be getting produced, but we know that much amount of that much huge volume of urine we are not excreting at all. So we are excreting 1 to 1.5 liters of the urine per day. So how this thing is becoming possible, though huge volume of the initial filtrate is getting produced, how we are excreting very little amount of urine? Okay, that we have to check. So in this first step, whatever glomerular filtration capillary pressure I told, okay, so here we will check what is the filtration membrane. So whatever blood is there, blood which is coming to the glomerulus region, glomerular capillaries, it is getting filtered through a 3-layered filtration membrane. So what are those 3 layers? Let's check here. So you will be getting 3 layers. One, that is capillary endothelium. This is a capillary endothelium, which is a fenestrated capillary. Fenestrated in the sense it is having number of slits or small openings over it. And then you will be getting the modified simple squamous epithelial cells. Those are known as podocytes. Podocytes, which are modified simple squamous epithelial cells of visceral layer of the Bowman's capsule. I told this is a previous year question. So, and in between this podocyte and the fenestrated capillary, you are getting the basement membrane. So whatever this particular podocytes are there, they will be wrapping in an intricate manner over the fenestrated capillary in such a way that you will be getting spaces here, and through this space, whatever blood is coming there, it will be getting filtered so finely in such a way that you will be getting the initial filtrate. Its composition is like plasma without protein. So as it is filtered very nicely, we could call this process as ultrafiltration. And what is there in the initial filtrate? Here I have written initial filtrate is nothing but plasma minus proteins inside it. Okay, and we know the composition of plasma also previously we have studied in the circulatory system. Plasma is a fluid part, fluid matrix of the blood, and plasma contains number of nutrients like glucose, amino acid, nutrients are there, important components are there, and huge volume of water is also there. Okay, so now whatever this initial filtrate is there, as I have written here, every minute 125 ml of initial filtrate is getting formed. In that way 180 liters per day. Whatever this particular initial filtrate is there, getting produced, getting stored there in the Bowman's capsular space. Now it will be passing through the tubules, means PCT, then loop of Henle and the DCT. And on the way, how it is going to become concentrated, that we have to check in the next two headings, that is reabsorption and the tubular secretion. So simply I will tell you here, reabsorption is the process of taking the nutrients back into the blood circulation. So this reabsorption process occurs in the tubules, various tubules, either by active transport or the passive transport. So please remember this point, glucose, amino acid and the sodium ion will be getting absorbed by the use of energy, that is active transport, while water by passive transport. In the same way, the one more step, that is tubular secretion. Tubular secretion in the sense to maintain the ionic balance, to maintain the pH, means acid and base balance of the body fluid, there is need to secrete some ions in the filtrate. Okay, so whatever that particular ions are there, they are H plus, K plus and NH3. These are getting secreted inside the filtrate to maintain the ionic balance of the filtrate. So how this process is occurring, we will check now. Now we will check the next step, that is reabsorption and the secretion simultaneously. Okay, so whatever that initial filtrate is there, now from that Bowman's capsule, it will be passing to the tubules. Okay, so it will be coming to the proximal convoluted tubule. Now we know what a proximal convoluted tubule is there. Okay, it is having a simple cuboidal epithelial tissue with microvilli inside it. Okay, so definitely whatever that dilute urine is coming, initial filtrate is coming. Okay, here I have written all the nutrients, then 70 to 80% of water and electrolytes, maximum all the things are getting absorbed here. Okay, and due to the microvilli presence, and the bicarbonate ions are also getting absorbed. Okay, you could call whatever absorption is occurring in the proximal convoluted tubule, which is occurring compulsory here, that particular absorption will be occurring. You could call it as mandatory absorption, or you could call it as obligatory absorption also. Okay, when these many components are getting absorbed here, to maintain the ionic balance and the pH, here again the same three components are getting secreted as a tubular secretion, H plus ion, K plus and NH3. Okay, now if you check the osmolarity of the filtrate present in the proximal convoluted tubule with respect to the blood plasma, we will understand its value is 300 milliosmol. It means the initial whatever the filtrate is there in this PCT and the blood plasma, they are isotonic to each other. Okay, now whatever this filtrate is there from the PCT, after maximum absorption of the water as well as the ions like sodium chloride also, it will be now moving to the next part. So from the cortex, from the PCT is present in the cortex region. Now that filtrate will be going to the next part, that is loop of Henle, which is there in the medulla region. Okay, so the filtrate will be passing now to the descending limb of loop of Henle. Okay, we will check what is the feature of descending limb of loop of Henle. So here I have written descending limb of loop of Henle is permeable for water, but it is highly impermeable for the electrolytes. It means from this region also there is a possibility of absorption of water, but there is no any absorption or any secretion is not possible with respect to electrolytes. Okay, so if you check here from the descending limb of loop of Henle, water is getting absorbed. Okay, so as water is getting absorbed from this particular region, filtrate will be going on concentrated. So here I have written the values. So you will be getting 400 milliosmol, then it will become concentrated as water is getting absorbed. Then from 600 to 800, and when it is coming to the end of descending part of loop of Henle, it is becoming almost 1200 milliosmol. highly concentrated. Now from here this filtrate will now ascend in the loop of Henle. Okay, ascending limb of loop of Henle is having a thin segment and then a thick segment. Okay, overall whatever this ascending limb of loop of Henle is there, it is permeable for the electrolytes like sodium chloride, and it is totally impermeable for the water. So definitely absorption of water is not possible from the ascending limb of loop of Henle, but electrolyte like sodium chloride is getting absorbed here. So here we could check now whatever sodium chloride is getting absorbed here from this particular region, okay, from the ascending limb. As electrolyte sodium chloride is moving out from this particular region, you could say water is there as it is, but sodium chloride is moving out. So you could say water filtrate is there. As it ascend in the ascending limb of loop of Henle, slowly it will be getting diluted. So right from the end of descending limb, if you check from 1200 to 900, 600, 400, and at the end of the loop of Henle, ascending limb of loop of Henle, the osmolarity will be 300 milliosmol. Okay, now whatever this filtrate is there from the ascending limb of loop of Henle will be going to the next part again, which is there in the cortex, that is in the distal convoluted tubule. I told you already, distal convoluted tubule is also having the simple cuboidal epithelial tissue, but there is no brush border. So, but then also here I have written absorption of water as well as the sodium chloride is possible here, but that is a conditional reabsorption. Conditional reabsorption in the sense in the presence of ADH, antidiuretic hormone, or you could call it as vasopressin. Okay, so based on the demand of the body, based on the need of the situation, based on that only, from this region either to absorb water or not, either to pass dilute urine or the concentrated urine, how much volume of water should go outside, that is decided here. So you could say in the DCT, water absorption and the sodium absorption, ionic absorption like bicarbonate ion absorption is possible, but it is a conditional absorption in the presence of ADH, antidiuretic hormone. In the same way, you could say tubular secretion is also occurring in this region. Same ions like H plus, K plus and the NH3 are getting secreted in that fluid. If you check there, as compared to the ascending limb, as the fluid will be moving to the distal convoluted tubule, it is again, its osmolarity is 200 milliosmol, means it got diluted. Okay, then decision is made. Okay, then when that particular filtrate will be coming down to the collecting duct. Collecting duct is a straight tubular structure which is starting right from the cortex. It is going till the medulla completely, and via that only urine is going. Okay, so whatever this collecting duct is there, from the collecting duct also considerable amount of water is able to get absorbed, but that is also in a conditional way in the presence of antidiuretic hormone only. Okay, from this region, okay, you have to remember one special point here, one important point. From the collecting duct, little amount of urea is getting inside the medullary interstitium, and this particular urea, whatever is coming in the medullary interstitium, it is helping to maintain the osmolarity high in the medullary interstitium. Okay, so I could say here in the collecting duct, water filtrate is there, its osmolarity, you could say it is hypertonic with respect to the blood plasma. So here, as I told you, mammals are having capacity to do the concentration of their urine. Okay, so how that concentration of urine is occurring? So just now I told you regarding how the absorption of water occurs in the PCT. Then when water moves to the descending limb of loop of Henle, again water reabsorption is occurring, and the water filtrate is there, it is getting concentrated. But as it moves up to the ascending limb of loop of Henle, again electrolytes are moving out and it is getting diluted, and in the diluted form only it is going to the distal convoluted tubule where the conditional reabsorption occurs. Okay, so here what will concentrated urine formation is occurring. So for that, what is that special mechanism? We could call that mechanism as countercurrent mechanism. So countercurrent mechanism, which is going on in the two components like in the loop of Henle and the vasa recta, is responsible for creating the concentration gradient in the medullary interstitium. and due to that only the absorption of water is possible from the different parts, like from the distal convoluted tubule. Okay, so what is that countercurrent flow means? So countercurrent flow, simply you could say, if fluid is flowing in opposite direction to each other, like for example you could say here, this is like inner water diagram I have drawn, this is the loop of Henle. So this part is descending limb of loop of Henle, where the filtrate is moving in downward direction, while what this part is there, this is the ascending limb of loop of Henle, where filtrate is moving in upward direction. So both of them, like in both of them, water filtrate is moving, it is moving in opposite direction to each other. So here you are able to see this is a counter current flow. In the same way, if you see this outer side, whatever I have drawn, that is a vasa recta. So whatever vasa recta is there, this is the descending limb of vasa recta. In the descending limb of vasa recta, you could say in the vasa recta blood is flowing. So in the descending limb of vasa recta, blood is moving in this direction, downward direction, while in the ascending limb of vasa recta, blood is moving in the upward direction. So anyhow, in both these particular vasa recta as well as in this particular loop of Henle, the movement of filtrate in the loop of Henle and the blood in the vasa recta is creating counter current, and anyhow both of them are in proximity, in the sense they are very close to each other, and due to this only the concentration gradient, means here concentration gradient is getting created in the medullary interstitium. Means the concentration gradient is created right from this cortex region. If you check, the osmolarity is getting increased right from 300 to 6 to 900. In this way it is going on increasing till 1200 milliosmol in the medullary interstitium for this counter current flow, which is running in both this particular loop of Henle and vasa recta, and as they are close to each other, this particular thing is helping to create the concentration gradient in the medullary interstitium, and it is only helping to pull out the water, especially from the distal convoluted tubule. So here they have given sodium chloride and the urea are playing important role in maintenance of this particular medullary interstitium gradient. So how? See here, we know descending limb of loop of Henle is permeable for water, so water absorption became possible. But how? So if you check here, you could understand what ascending limb of loop of Henle is there. It is permeable for electrolytes, but not permeable for the water. So from this region, sodium chloride is moving out. So whatever sodium chloride is moving out from the ascending limb of loop of Henle is taken by the descending limb of vasa recta. Okay, and whatever little amount is there, it will be there in the medullary interstitium also. And see here, sodium chloride got absorbed. So the concentration gradient is going on increasing in this way. But as this blood is flowing through the ascending limb of vasa recta, slowly sodium chloride is coming out again from the ascending limb of vasa recta. So here simply, sodium chloride comes out from the ascending limb of loop of Henle, is taken by the descending limb of vasa recta, and again it is coming out in the medullary interstitium from the ascending limb of the vasa recta. And due to this, whatever high concentration is getting created here around the descending loop of Henle, due to this reason only, water absorption is becoming possible. In the same way, water urea is there, urea which is coming out from this particular collecting duct, it is also creating the high concentration in the medullary interstitium, and due to that only, it is becoming possible for absorption of water from the collecting duct, and in that way concentrated urine will be getting produced. Okay, now here we are going to check the micturition process. Micturition is the process of passing out the urine. So we know once the urine got concentrated, it will be coming into the renal pelvis, and from that renal pelvis it will be coming into the ureter. Via ureter it will be getting inside the urinary bladder. When urinary bladder will be getting full of the urine, that time you could say whatever stretch receptors are there in the wall of urinary bladder, they will be getting stretched. Due to those stretch receptor, due to the stretch, the signal will be going, a signal is getting initiated which will be going to the central nervous system. Now in response to that signal from the stretch receptor, central nervous system will be sending motor signal to the smooth muscles which are present in the wall of urinary bladder for the contraction, and in the same way it will be sending signal to the sphincters which are present in the urethra region for the relaxation process. And when contraction of urinary bladder occurs and the sphincters present in the urethra get relaxed, the urine will be released outside, and this process is known as micturition. Now we will check the regulation of kidney function. So whatever kidney's function is there, it is regulated by three main structures, you could say. One is hypothalamus, second is a juxtaglomerular apparatus, and the third one is heart up to one extent. Okay, so how this function is getting regulated? See, so what is hypothalamus is there? We know thermostat of our body. Okay, so hypothalamus will be getting, so hypothalamus, how it is controlling, what is that condition when it is doing the work? Okay, so when we suffer from the dehydration or much loss of the water is happening from our body, you could say during that condition whatever osmoreceptors are there in our body get activated. So in response to loss of the body fluids, osmoreceptors will get activated, and the osmoreceptors will be sending the signal to the hypothalamus, and then hypothalamus will be releasing its hormone, that is vasopressin or antidiuretic hormone. Okay, and we know antidiuretic hormone is helping for the conditional reabsorption of water from the distal convoluted tubules. So the water absorption will occur from the distal convoluted tubule, and whatever loss of water occurred, it will be getting stored. So diuresis will be stopped, and once diuresis got stopped, water levels come in a proper way in our body, automatically the osmoreceptors will be getting switched off. Okay, so here the role of ADH also we could understand. ADH is a vasoconstrictor, and it will be helping to increase the blood pressure, thereby the glomerular filtration rate also. So you could remember ADH is a vasoconstrictor. Okay, now the second mechanism, that regulation is done by juxtaglomerular apparatus, JGA we call it as. And what exactly this juxtaglomerular apparatus? It is a sensitive area formed when two structures, one that is afferent arteriole and other that is DCT, come in close contact with each other. That time some modification occurs in the cells and a sensory area is getting created. You could call it as JGA apparatus. Okay, so when this JGA apparatus is getting activated, so the condition for its activation, you could say, when there is fall in the glomerular filtration rate, means GFR happens, that time this JGA region will get activated, and it will be releasing one enzyme known as renin. Only one N is there, don't get confused with double N which is there in the digestive system. Okay, and you could call it as angiotensinogenase enzyme also. This enzyme is having capacity to do the conversion of inactive form, that is angiotensinogen, into angiotensin 1. Again, in turn, it will become angiotensin 2, which is a powerful vasoconstrictor. Okay, so whatever angiotensin 2 is there, which is powerful vasoconstrictor, due to the vasoconstriction, blood vessels constriction will occur, due to which the increase in the blood pressure occur. And when blood pressure is increasing, automatically glomerular filtration rate will become more. So the condition is what? Fall in the GFR, and it will be solved by the increase in the glomerular filtration rate. Okay, now one more thing here I want to explain. What is this angiotensin 2 is that here I have written angiotensin 2 VC, in the sense it is a vasoconstrictor. It's powerful vasoconstrictor. It will do two things. In this way it will increase the glomerular filtration rate also, and it will activate the adrenal cortex also, and by that aldosterone will be getting released. And aldosterone, you could say, will be helping for the absorption of sodium and water from the distal convoluted tubule. So overall, whatever renin, angiotensin and aldosterone are working together, you could call this system as RAS system. renin, angiotensin, aldosterone system. Okay, now the final work of the control over the kidneys function is done by heart up to one little extent. So when this work happen, actually heart will be releasing one factor that is known as atrial natriuretic factor. Atrial natriuretic factor. So what is that condition? Okay, again whatever this atrial natriuretic factor is there, it is vasodilator. We have done here the ADH as well as angiotensin 2, which are vasoconstrictor, but here you could say whatever ANF is there, it is the vasodilator. Okay, so when more flow of blood occurs to the atria of the heart, that time you could say this ANF, atrial natriuretic factor, is getting released from the atria, which is acting as a vasodilator. Thereby there will be a decrease in the blood pressure occurs. Okay, so here you have to understand whatever this atrial natriuretic factor is there, it is acting as a check on the RAS mechanism. RAS mechanism which is helping to increase the blood pressure, whereas the atrial natriuretic factor is helping to decrease the blood pressure. So here by the three components, you could say hypothalamus, juxtaglomerular apparatus and the atrial natriuretic factor, the control over the kidneys function is done. So now finally we will check the urine composition, the pH and color. So whatever urine is getting excreted, we know it is a concentrated urine having its pH around 6. Color is yellow due to a special pigment inside it, that is urochrome. Around 25 to 30 grams of urea is getting excreted from our body per day. Okay, and from the this particular urine detection, urine diagnosis will give us idea regarding any conditions like diabetes mellitus. So especially in the sugar patient, we could say we will be getting ketone bodies present in the urine as well as the sugar, okay, glucose in the urine. Okay, that is indication of diabetes mellitus. Okay, now other than kidneys, some another structures are there, organs are there in our body which are also helping to do the excretion of some another waste materials from our body. So what are they? Let's see here. So they are lungs, liver, sweat gland, sebaceous gland. Okay, and see here, whatever lungs are there, we know that lungs are helping to remove the CO2 continuously from the body. So each minute 200 ml, okay, of the CO2 is removed from the lungs. Apart from that, little amount of water every day is removed by the lungs. Liver is helping to remove the bile containing bile pigments, you could say, bilirubin, biliverdin, some hormones, drugs, vitamins, okay, from this particular liver. But all these things will be generally going out via the digestive system. Sweat gland, we know its main function is to do the evaporative cooling, maintaining the body temperature. But apart from that, whatever sweat will be there, it will be having a fluid with sodium chloride, very little amount of urea and lactic acid also inside it. Okay, so and finally sebaceous gland, which will be secreting oil actually and giving a protective covering to our body. It is also removing little amount of waste material like hydrocarbons, wax and the sterol. Okay, last they have given a small point that whatever our saliva is there, through saliva also very little amount of nitrogenous waste is removed. Okay, now the final thing we are going to check the disorders. Okay, so very few disorders they have given. One is the renal caliculi, means generally we call it as kidney stones, which is a crystallized stone of the oxalates. Okay, glomerulonephritis, itis means inflammation. Inflammation in the sense rubor, tumor, calor, dolor, all these immunological terms will come there. So the inflammation of the glomeruli of the nephron is known as glomerulonephritis. Then the last one, that is uremia. If kidneys are not functioning properly, then urea is not excreted by the kidney properly. So the excess of urea will be getting accumulated in the blood and it leads to a condition that is known as uremia. It will be a very harmful condition. So there is need to remove that excess of urea from the body by using some artificial methods. You could call it as artificial kidney, or you could call that process as dialysis. Okay, so what is there in the dialysis? We will check. So in the dialysis, first of all, you could say blood is taken out from a convenient artery. Blood is taken out from a convenient artery, and whatever that blood is taken out, it is added with an anticoagulant, that is heparin. whatever that blood is there, it is allowed to flow through a coiled circuit made up of cellophane. Okay, and when blood is flowing through that particular cellophane made coiled structure, which is known as artificial kidney, also here I have written, that cellophane is surrounded by, actually around it, the dialysate is there, dialysing fluid is there. Dialysing fluid is what? It is a fluid whose composition is like exactly like the plasma, except the nitrogenous waste, that is urea inside it. Without urea, leftover composition is there in that dialysing unit. So dialysing fluid, you could say, the dialysing fluid will be flowing around this particular artificial kidney. So what will happen, whatever blood is flowing through this particular dialysing unit or artificial kidney, as surrounded by that, you are getting that particular dialysing fluid, as you don't have any type of urea inside it. From that particular from our blood, okay, whatever urea is there, it will be moving inside the dialysing unit, dialysing fluid, it will be taking that urea, so that blood will be getting purified, it will be getting free from the urea, and again that blood first of all added up with the anticoagulant, means antiheparin, and then that blood is allowed to go via a vein to the body back. Okay, so this particular process is used if a person is suffering from uremia. Okay, but if kidneys are getting failed only, means they are not able to do the function, you could say it is an acute renal failure. So in this condition, there will be only one option, that is a kidney transplantation. So kidney, by the time of kidney transplantation, first of all donor is selected. So donor, if a monozygotic twin is there, it will be the best option. But apart from that, if any close relatives is there, from the close relative generally the organs are accepted. Especially the kidney transplantation is done from the close relative. Okay, why from the close relative it is done? Because we want to avoid the rejection by the immune system. Okay, only one kidney is transplanted because one kidney will be sufficient to do the work. Okay, and whatever person who have undergone this transplantation, kidney transplantation, that person have to take the immunosuppressant drug throughout their life to avoid the rejection of this kidney by the immune system. I hope this video will be definitely helping you, because overall complete lesson I have covered in the two parts, part 1 and part 2. Okay, I hope this is better for you. Thank you. Thank you everyone for supporting me and my channel. Please don't forget to like, share and subscribe my channel, and please write a comment in the comment box below. Thank you.